Hello everyone, I'm Shweta doing second MSc Microbiology. We are going to discuss about blast of rice. First, the introduction. Rice blast disease is caused by Magna porte oryzae and Pyricularia oryzae. It is one of the most damaging diseases of rice. This disease was first known as rice fever disease in China and it is identified yearly as 1637. Later, the disease of rice blast disease was first recorded in 1704 in the place of Japan and it reported from 85 rice producing countries or regions worldwide and it can also destroy rice that feed for 60 million peoples. General characteristics of rice blast fungi. It belongs to Ascomycetes fungus and it can reproduce both in sexually and asexually. It can survive in seeds, plant residues and weeds. Optimum relative humidity 90%. Optimum temperature for rice blast fungus 20 to 25 degrees Celsius. It produces the toxins like pyrrolidine and alpha picolinic acid. This is the microscopic view of rice blast fungi. The disease cycle of fungus. The rice blast pathogen overwinters as fungal strains or spores on seeded rice stubble, on infected seed, or on living plants, such as grass weeds that grow over winter. Next, the infection in a new season may originate from the fungus overwintering on rice straw or may come from diseased grassweed species within or around the crop. Rice blast spores are transported by wind and water, often over long distances, and can infect rice plants after landing on them. Many infection cycles may occur within a cropping season if weather conditions are favorable. Warm temperatures are around 25 degrees Celsius and high humidity greater than 90% at night favor infection. Symptoms of the blast fungus on rice crops. Rice blast spores can infect plants at all growth stages from seedling to maturity. Symptoms develop on all above ground plants. Lesions or spots are most common symptom. Lesions are usually 1 to 1.5 centimeter long and 0.3 to 0.5 centimeter wide. Crops under stress may exacerbate symptom development leading to rapid growth decline, especially in susceptible varieties. The first symptom, leaf blast. Leaf lesions start as small white, gray, or blue tinged spot on the crop. Under moist conditions, lesions enlarge quickly to either oval or diamond shaped spots or to linear lesions with pointed ends, gray or white centers, and narrow brown borders. Severe infections may lead to death of leaves and old plants. It provides inoculum for panicles to become infected. This is the picture of a leaf blast on rice crop. The second symptom is collar rot. If a rice blast lesion is located at the junction of the leaf blade and leaf sheath, the entire leaf can be killed. The leaf collar lesion discolors to brown and the leaf blade dies. This is the infected plant with collar rot. The third one is node infection. Infected nodes appear black to brown and dry. An infection at the node often results in the stem breaking. This is the node infection in premature rice crop. The fourth one is neck rot. Neck rot may result in death of the entire panicle. Symptoms appear at the base of the panicle starting at the node. The tissue turns brown and shrivels, causing the stem to snap and lodge. The crop's head slides down when infected with the neck rot. And finally, panicle blast. Panicles which do not break or fall off as a result of neck rot may turn white to gray. Partially, it is infected panicle may show gray-brown lesions among the panicle branches and on the stems of the florets. The florets which do not fill turn gray. This is the panicle blast on rice crops. The favorable conditions for the rice blast to grow: upland rice environment, presence of the blast spores in the air throughout the year, cloudy skies and frequent rain and drizzles, high in nitrogen levels and relative humidity, and low soil moisture and high leaf wetness or dew period. The control of rice blast fungus on rice crops. Commonly used blasticides are isoprotheolane, probenzonazole, pyroquinone, tricyclazole, etc. And the broad spectrum fungicides are azoxystrobin, the first spray used during tillering stage and the second spray during flowering stage, and finally the seed treatment which are used tricyclazole or beam 75 WP 4 gram per kg seed. The biological control or Pseudomonas fluorescens strain PF1. Thank you for your patience.